This is the case study Which Little Piggy Went to Market, Bioinformatics and Meat Science by Emma and Navi. We will be going over two questions in this presentation. Question 12, which asks, what do you conclude from figure 1, which is seen here, and why were lanes 1, 2, 3, 5, and 6 included in this experiment? So first of all, let's go over figure 1 in greater detail and determine what is present in each lane. So in lane 1, we have our 123 base pair DNA ladder, and this can also be called a molecular weight ruler. And what this does is we can use it as a reference in order to determine the size of our controls and unknowns. And as we can see, a band has formed at 123 base pairs, 246 base pairs, and 369 base pairs. In lane 2, we have one of our negative controls in which no DNA is present. Since no DNA is present, the primers have nowhere to bind and no DNA amplification can occur because no DNA is present to begin with. So as a result, in lane 2, no band has formed, which makes a lot of sense. So in lane 3, we have another one of our negative controls in which no primers are present. So even though there's still DNA located in lane 3, it can't be amplified because DNA amplification can only occur in the presence of primers and when primers bind to the DNA. So that's why in lane 3, no band is present as well. So in lane 4, we have our positive control, which is boar DNA. So we know that boars are male and that on their Y chromosome they have the SRY gene. And since the SRY gene makes up the DNA sequence that we're using in this experiment, the specific primers are actually able to bind to the DNA and amplify it. So this allows PCR to proceed, and it produces a visible band at 158 base pairs. So we can see that band right here. Now in lane 5, we have another negative control which contains sow DNA. We know that sows are female and that they contain 2 X chromosomes. And we also know that they do not contain the SRY gene or a Y chromosome. So because of this, the primers present can't bind to the DNA sequence because the DNA sequence is from an SRY gene and they do not contain that gene. So because of this, no DNA amplification can occur, and that's why in lane 5, no band is produced. And finally, in lane 6, we have our unknown sausage sample. And we can see that, yes, although it's very faint, we do indeed see a band produced at the same location as our positive control, which is the boar DNA, at 158 base pairs. So we'll go into what that means on the next slide. So what were we ultimately able to conclude about figure 1? Well, we were able to conclude that yes, the unknown sample, which is located in lane 6, did in fact contain boar meat. And we know this because, like I said previously, the bands are both located at 158 base pairs. And so lane 4 contained the boar sample, and lane 6 must have to contain boar meat as well since they're both located at the same location. And so this means that the SRY gene was in fact present in the unknown sample and that DNA was replicated as a result, and therefore we know that yes, boar meat was contained in the unknown sample.